Good morning, all. Welcome. I would like to welcome you to the first presentation of the HIAAD Basic Science Seminar Series and the first satellite event of Genetics 2022. For those who are not familiar with it, Hellenic Initiative Against Alzheimer's Disease, or HIAAD, is an initiative started about 1 year ago, whose major goal is to implement a Greek National Dementia Action Plan to fight Alzheimer's disease using a range of approaches, one of which is the promotion of research in the field. HIAAD brings together from Greece and abroad people with different backgrounds interested in fighting Alzheimer's disease, such as doctors, researchers, educators, entrepreneurs, caregivers, and business people. It's coordinated by the Precision Medicine Center of Excellence for Alzheimer's Disease and Johns Hopkins in the United States, and the Bioinformatics and Human Electrophysiology Laboratory at the Ionian University. A central objective of HIAAD is to promote basic science research in dementia and Alzheimer's disease in Greece by stimulating and expanding research collaborations. We hope to achieve this in part by organizing this monthly seminar series with invited speakers to exchange ideas and promote discussions and collaborations on AD research. So our first speaker today is Dr. Dimitris Kapogiannis, who is a clinical investigator and chief of the Human Neuroscience Unit at the National Institute on Aging, and adjunct associate professor at the Department of Neurology and Johns Hopkins University. Dr. his medical degree from the National and Kapodistrian University of Athens in Greece and completed a neurology residence program at the Massachusetts General and Brigham and Women's Hospitals at Harvard University, followed by a fellowship in cognitive and behavioral neurology at the National Institute of Neurological Disorders and Stroke at the NIH. His research program includes translational biomarkers and clinical studies on cognitive aging and various neurodegenerative diseases, with a primary focus on Alzheimer's disease. He has been a pioneer in isolating exosomes enriched for brain cell origin from blood and using them to derive biomarkers for neurological and psychiatric diseases and as outcomes for experimental treatments. The title of his presentation today is Exosome Biomarkers and Precision Medicine for Alzheimer's Disease and Beyond. Dimitri, thank you very much. And I would like to welcome you, and the floor is yours.
Vaso, thank you so much for this kind introduction and for the kind invitation to present in this conference series. It's humbling to be the first. I hope that it will be a series that will excite all of us. Actually, correction, the first was Yannis last month, who already talked to us about exosomes, and so I'm following on his footsteps. So good morning, everybody, and it is great to feel that I am among friends. And without further ado, I will jump into it. The pathogenesis of Alzheimer's is complex. The attention of the field has been captured by its most obvious pathologic hallmarks, extracellular plaques containing misfolded amyloid beta and intracellular tangles containing misfolded tau. This has drawn attention for decades, if not for more than a century, and perhaps it has obscured other processes that are equally important. The disease is characterized by numerous additional abnormalities at the biochemical, cellular, and tissue levels. The disease spreads from affected to unaffected regions through transcellular propagation of A beta and tau. Reactive astrocytes and microglia attempt to isolate the initial insult and repair it. But unfortunately, the ensuing neuroinflammation, it's often counterproductive and results in further damage. Along these lines, there is also insulin resistance, decreased metabolism of glucose, and mitochondrial abnormalities, And these factors create an energy crisis for neurons and astrocytes at a time when they would actually need an abundance of energy to repair the insult. Also, the background, there is impaired autophagy and lysosomal dysfunction that precludes successful recycling of misfolded proteins and damaged organelles. The mitochondrial abnormalities are also resulting in oxidative and also more broadly cellular stress that leads to lipid and DNA damage and against the background of impaired cellular stress responses. These processes damage synapses and result in synaptic dysfunction and synaptic degeneration, and eventually axonal and neuronal degeneration. This is obviously a very complex series of events. And in my lab, we have been using circulating extracellular vesicles to characterize these biochemical and cellular abnormalities of Alzheimer's, as well as Parkinson's and other neurodegenerative diseases in living individuals as a form of liquid biopsy. Brain cells, neurons, astrocytes, microglia, they all secrete extracellular vesicles as part of their normal and pathologic function. Brain extracellular vesicles have been shown to cross the blood-brain barrier through transcytosis and other mechanisms that are not well understood today, and can be detected in peripheral blood. Our lab has pioneered the methodology for isolating brain-derived EVs by selective immunocapture. We have isolated subpopulations of neuronal EVs by targeting neural cell adhesion molecules such as L1-CAM, NCAM, and NCAM, and astrocytic EVs by targeting neuronal markers, by targeting astrocytic markers such as GLAST, which is an aspartate glutamate transporter. The term extracellular vesicles refers to vesicles generated and released by cells through a variety of mechanisms. The main types of EVs are exosomes that are derived for the late endosome multivesicular body, and they are sorted to the membrane as exosomes. And also microvesicles were derived by direct budding of the plasma membrane. These vesicles form a continuum of sizes, cargos, and molecular composition. Circulating EVs as a whole effectively sample cargo from almost every cellular compartment of neurons, astrocytes, or cells of origin in general. From the lipid bilayer, which samples the membrane, the plasma membrane, as well as intracellular membranes, especially the endosomal membrane, to various transmembrane proteins on the surface. Some of them are constitutive components of exosomes, such as, or microvesicles, such as the tetraspanic CD81, CD9, CD63, various cell adhesion molecules, various transporters, to various intracellular signaling mediators, to RNA and DNA species, to enzymes, to pathogenic proteins such as tau and A-beta, or alpha-synuclein and TDP43, to whole fragments of organelles, such as probably mitochondrial fragments. This diverse cargo provides a wealth of possible biomarkers for diagnosis, prognosis, and detection of therapeutic response. The very first study of neuronal EVs focused on ATN, amyloidosis, tauopathy, and neurodegeneration to derive biomarkers for Alzheimer's disease. For those of you that you're not familiar with this nomenclature, there is a current approved organizational schema for defining biomarkers of Alzheimer's disease, which is called the ATN schema, which stands for amyloidosis, tauopathy, and neurodegeneration. Specifically, we conducted a case-controlled study focusing on the stage of mild cognitive impairment, early dementia in the continuum of Alzheimer's progression. And we demonstrated that AD patients compared to controls had higher levels of phospho-tau in their neuronal exosomes, both phospho-181 tau and phospho-396 tau compared to controls, as well as A-beta-42, and at trend levels of total tau. These differences were large and were able to discriminate between AD patients and controls with high accuracy. The findings of this study have been validated many times by us and others, and have launched pretty much the neuronal EV field. The field of biomarkers based on neuronal EVs, to be accurate. Here is a recent replication, a study that was published in Alzheimer's and Dementia, where this group demonstrated that any stepwise increase of phospho-tau, total tau, and A-beta-42 from controls to MCI to AD using the same methodology as we first demonstrated. Moreover, besides ATM, we've been attempting to introduce new domains to the biomarkers field for Alzheimer's. And now I would like to turn our attention to neuronal insulin resistance, which in my mind is an important and often overlooked aspect of disease pathogenesis that may explain the epidemiologic association of Alzheimer's with diabetes. Over the years, we have detected multiple insulin signaling cascade mediators in neuronal EVs, which opened the way to using them as biomarkers to interrogate the status of insulin signaling in parent neurons. A brief primer on the insulin cascade. Insulin binds to the insulin receptor, and from there activates a cascade through a series of activating phosphorylations. The first node is this molecule called IRS1, insulin receptor substrate 1, that can be activated by tyrosine and inactivated by serine phosphorylations. Therefore, if tyrosine phosphorylations predominate, the cascade proceeds downstream, and there are two branches. One is the so-called canonical pathway that proceeds through PI3K to AKT, and from there to GSK3 beta, all the way down to mTOR. There is also an alternative MAP kinase cascade that is more related to neuroinflammation, as we believe. Insulin resistance means decreased cellular responsiveness to insulin, therefore decreased propagation of signals along this one or both of these cascades. And this can be reflected at the first node through increased p-serine IRS1, since these are inhibitory phosphorylations, or increased p-tyrosine IRS1, since they are permissive phosphorylations, or an imbalance between p-serine and p-tyrosine phosphorylations. So how did we determine that this whole complex cascade mediators could be biomarkers? Well, it all started with a discovery study where we were assessing the proteome of L1-CAM EVs, the neuronal EVs, and comparing it with the proteome of total EVs and epithelial origin EVs as controls, and where we saw that multiple of these molecules existed in high levels and at different phosphorylation sites. A clinical pathologic study, which is often where we draw our inspiration by Conrad Talbot and colleagues, had already established that p-serine IRS1 which is a common marker of insulin resistance in peripheral tissue, also shows a dramatic increase in the brains of individuals with MCI and Alzheimer's. We followed up this clinical pathologic study with a neuronal exosome study, and we examined whether a similar increase in p-serine IRS1 exists in neuronal EVs, mirroring what happens in tissue. And indeed, we found that neuronal EVs of AD patients compared to controls show elevated levels of p-serine IRS1, whereas total levels of IRS1 are unchanged. Therefore, already by 2016, we had performed this series of small to medium-sized studies, case control studies, and had several candidate EV biomarkers for Alzheimer's. Biomarkers reflecting ATN, amyloidosis, A-beta-42, tauopathy, in form of these phosphomolecules, phospho-tau molecules, and neurodegeneration, better reflected by total tau, as well as insulin signaling mediators. All these studies had been performed in clinical AD compared to control. Nevertheless, we didn't lose view of the sight that a more desirable goal is to actually have biomarkers for preclinical disease, because that is the time when, if you detect the disease, you can apply primary prevention interventions and prevent the cascade from evolving. Therefore, we undertook large-scale validation studies using longitudinal cohorts, the Baltimore Longitudinal Study of Aging and the Wisconsin Registry for Alzheimer's Prevention, and focusing on preclinical rather than clinical AD. And these studies were published in the last couple of years in JAMA Neurology and Alzheimer's and Dementia. The study, leveraging samples from the VLSA, is worth detailing. The VLSA, it's a longitudinal study where various individuals has been going on since the 50s up until now. I should have updated now to actually 2021. And individuals enroll at different times in this study when they're cognitively healthy. Over time, some of these individuals develop Alzheimer's disease. We mark them here as red. We went and identified when a certain individual, who was previously cognitively normal, developed Alzheimer's, and we marked this visit as the index visit, the visit of the first clinical manifestation of Alzheimer's disease. Then we went back to our freezers and we pulled samples from the time when this individual was still normal. We also identified super controls, that is, individuals who enrolled in the VLSA and remained cognitively normal for the entire length of their participation, which is either death or withdrawn. Most often, people remain in the VLSA until they're very old. The final data set included 887 samples from 350 individuals, 128 of whom developed Alzheimer's disease at some point, and 222 of whom remained cognitively normal for the entire length. This interval between the first manifestation of Alzheimer's and the earliest preclinical visit was 3.3 years of average, but in some people went back to even 9 years. Therefore, this is what we are contrasting. We're contrasting cognitively normal individual who at some point will go on to develop Alzheimer's, and cognitively normal individual who will never develop Alzheimer's. This study confirmed the previous findings regarding phospho-tau and phospho-IRS1 biomarkers. Specifically, we found that P231 tau and especially P181 tau shows large elevations and highly significant in future AD individuals compared to future control subjects. Moreover, both P-tyrosine and P-serine IRS1 show smaller but significant differences between future AD patients and controls. A model combining longitudinal data for phospho-tau, tau, and IRS1 achieved an area under the curve in discriminating between these two groups by almost 90%. Moreover, this study generated additional exciting findings regarding the relationship of these EV biomarkers with cognitive performance across all individuals, both controls and future AD. For instance, we found that phospho-tau, phospho-181 tau, and P-serine IRS1 showed negative correlations with multiple cognitive domains, including verbal memory. In particular, well, the association between phospho-tau and cognition is something that everyone would expect, but the very strong correlation between p-serine IRS1 and verbal memory is not something that everybody readily expects. And this is a reflection of the study by Talbot, where he had shown that p-serine IRS1 levels in the cortex of diseased individuals show indeed a negative correlation with their cognitive performance when they were still alive. So here we see a replication of a clinical pathologic study finding using neuronal EVs. This is basically what we mean when we say with exosome biomarkers, we can arrive at something akin to liquid biopsy. Moreover, we were very keen to conduct studies combining EV biomarkers and neuroimaging. In this first study of its kind, we showed that p-tyrosine IRS1 is associated with increased MRI volume, increased gray matter volume in individuals with Alzheimer's disease. Interestingly, this association was not a random collection of voxels. This association closely reflects the pattern of IRS1 gene expression, as seen here in the Allen Brain Atlas. For those of you not familiar with Allen Brain Atlas, is a publicly available and wonderful resource released by the Allen Institute, where they have done microarrays in tiny areas of the cortex throughout the brain. And they have quantified the gene expression for every gene. And this is a common way of visualization. So if you observe here, this is something that emerged without hypothesis in a data-driven way from our VBM analysis. And the pattern of association between neuronal EVs and brain volume closely resembles what we see in the gene expression of IRS1 in the brain. This led to a hypothesis that enhanced insulin signaling is neurotrophic and promotes larger brain volume, but not in a random way, in areas where IRS1 is actually present and where insulin signaling may be expected to play a big role in determining neuronal homeostasis and neuronal trophism, which is the areas where IRS1 is present. We further tested recently this hypothesis in a study leveraging another longitudinal study cohort called ARID, the Atherosclerotic Risk in Community Studies. And we found that indeed p-tyrosine IRS1 is associated with temporal lobe gray matter. But not only IRS1, this time we looked both upstream at the insulin receptor and insulin IGF-1 receptor, as well as downstream at the levels of phospho-AKD, phospho-GSK3 beta, and phospho-P70S6K. And we found concerted associations with all these insulin signaling mediators and temporal lobe volume. Moreover, we looked at something bad, the white matter hyperintensity seen on MRI on these patients. And we saw that while insulin signaling mediators were associated with gray matter volume, positively associated with gray matter volume, they were negatively associated with the volume of white matter hyperintensities. Recently, we have had some exciting findings in Parkinson's disease. Specifically, we've analyzed, and this is unpublished data, so please keep them confidential, but I'm happy to share, we analyzed a pretty sizable cohort of PD individuals, 103 of whom are with normal cognition, 84 with mild cognitive impairment and 39 Parkinson's patients with dementia, as well as controls, and they're well matched. And we examined a series of hypotheses. I'm not going to be comprehensive. We measured more things and have more findings than what I'm going to show you. But actually, one of the hypotheses we wanted to test is whether Alzheimer's type pathology exists in these Parkinson's patients and whether it may explain cognitive decline. And indeed, we found that phospho-181 tau is increased in PD compared to control individuals as a group. And if we break it further down, the phospho-181 tau is increased in the PD individuals with impaired cognition, whereas PD with normal cognition is similar, have similar levels to controls, and seems to show a stepwise increase in MCI, especially a big jump, and same levels with MCI in PD dementia. This suggests that the emergence of cognitive dysfunction in PD patients may at least be partly attributable to the coexistence of AD pathology. I saw my friend Kostas Vikrelis in the background, so he may be interested in noting that. Moreover, we found that neuronal EVs of PD patients as a group have lower levels of p-tyrosine, IRS1, compared to controls. And this again concerns PD MCI and PD dementia patients. And very excitingly, we found that p-tyrosine IRS1, this is the good IRS1, to make it pretty plain, is negatively associated with MDS UPDRS Part 3, which is a scale that shows motor symptom worsening in Parkinson's disease. Moreover, insulin signaling phosphoproteins are emerging as biomarkers in psychiatric diseases. In a study conducted in collaboration with German collaborators, we found that activated phosphoproteins in the insulin signaling pathway, normalized by their total protein levels, so consistently lower levels in drug-naive first episode schizophrenia patients. This further implicated decreased insulin and mTOR signaling in schizophrenia. And very recently, a group without our involvement used L1-CAM EVs to derive p-serine IRS1 and showed that women with depression have higher levels of p-serine, the bad insulin resistance type of IRS1, in patients with major depressive disorder, and whereas there were no significant differences in men with major depressive disorder. This is a pretty big deal because not many biomarkers of blood biomarkers of depression exist, and points to an interesting sex difference. These and some other recent studies firmly introduced, I believe, neuronal EV biomarkers in the field of psychiatry. Now, the association between neuronal EV biomarkers of insulin resistance and disease traits and states opened the way for using them as, or at least examining whether they could be used as surrogate outcomes in clinical trials. And in studies spanning disease entities and interventions as diverse as diet, exenatide, which is an anti-diabetic drug, intranasal insulin, and anti-TNF-alpha antibodies, we have shown that these EV biomarkers can be used to show target engagement and predict response to treatments targeting the insulin cascade, akin to a messages in a bottle, according to a recent editorial. I will refer in more detail in a study, a secondary analysis of the exenatide PD trial, which was conducted in collaboration with investigators from the University College London. We performed a blind secondary analysis of plasma samples from a randomized controlled trial, which was conducted in PD individuals, which was, by the way, a positive study that showed benefit of 48 weeks of exenatide treatment on motor function. And the original study was published in Lancet. Therefore, it was a pretty big deal for the Parkinson's field. We were able to show blindly an increase in P-tyrosine activating IRS1, which is now even more notable since a few slides ago I showed you that this molecule is decreased in Parkinson's disease individuals. And here we show that the exenatide treatment is able to elevate it over time. And this elevation persisted even after washout at 60 weeks. The same type of elevations were seen all the way down in the canonical insulin cascade, mediators in the canonical insulin cascade, whereas there were no differences in the MAP kinase cascade. So therefore, we were able not to show biomarker response to exenatide, which matched the clinical response, and to point down the mechanism of this response into the canonical insulin signaling cascade. Recently, we analyzed samples from a clinical trial of infliximab. Infliximab is a monoclonal antibody that targets TNF-alpha, an inflammatory cytokine. And this trial was conducted in individuals with severe bipolar depression. The basis of this trial was evidence that at least some patients with bipolar depression have peripheral pro-inflammatory biomarkers and pro-inflammatory risk factors. Therefore, investigators in Canada assessed whether giving infliximab to bipolar depressed patients could alleviate their depression symptoms. Similarly to the previous study, we analyzed blindly plasma samples, and we tested a series of hypotheses to determine the mechanism of action of this response. Now, there are two potential pathways. Infliximab blocks soluble TNF-alpha binding and decreases in particular TNFR1 signaling. This shifts the cascade towards activation of another receptor called TNFR2, and which is considered neuroprotective. And we were able to confirm this hypothesis and to show that indeed treatment with infliximab resulted in consistent elevations in the canonical MAP kinase insulin signaling pathway, shown here by two representative members of the cascade. And moreover, this pathway is expected to activate JNK. We saw it indeed activating, and a downstream signal from JNK is activation of p-serine 312, and that is exactly what we found. Therefore, there is another example how we can use neuronal EVs to pinpoint to the exact mechanism of engagement in a clinical trial. Now, a few words about precision medicine. The multiple mechanisms involved in Alzheimer's, Parkinson's, and other neurodegenerative diseases, and the different timelines for their emergence may be reflected in disease subgroups. There may exist subgroups with prominent neuroinflammation, which will respond better if you target them with an anti-inflammatory treatment. Or there may be groups that have predominant metabolic abnormalities, or groups that they have a particular aggressive form of tauopathy, or a particularly aggressive amyloidosis. Currently, when we conduct trials, we subject all of these people to the same treatment, and we fail to see an effect. Wouldn't it be better if we were able to target our treatments to subgroups that have the relevant pathophysiology? This is exactly the goal of precision medicine, to target treatments to individuals that have a particular endophenotype. Therefore, The goal is to separate these people. How do you separate them, though? They don't have a label on their forehead saying, I belong to neuroinflammatory subgroup or to a tauopathy subgroup. Obviously, the way to do that is by biomarkers. But what evidence do we have that our biomarkers are able to discriminate between individuals that belong in the same diagnostic category? So far, I have been showing you group differences. Group differences are good for science and for statistics, but they mask individual trajectories. It's a different mindset when you want to develop precision medicine biomarkers than when you want to generate findings that refer to the entire disease. Now you have to pay attention to individual trajectories. And what evidence do we have that EV biomarkers may reveal disease subtypes? In the Baltimore longitudinal study, we had available repeated samples and a large N, which gave us the opportunity to examine changes over time in individuals. And here I show you the individual trajectories, and you see how complex the pattern is when you look at individual trajectories. You can readily appreciate a blueness, which is normal people, which is lower, and the redness, which is higher phospho-181 tau in future AD individuals. But there is a considerable overlap. We can try to make sense of this by looking at individual trajectories, at least by breaking them by age brackets. And these are these lines that you see here. These are trajectories averaged by age bracket. And it becomes apparent that phospho-181 tau starts to diverge in future AD patients compared to forever normals after age 70. And those that will end up at some point develop Alzheimer's disease started showing elevated phospho-181 in their neuronal exosomes. The opposite, interestingly, is true for phospho-231 tau, which is maximally different in younger age groups and starts converging at older age groups. In contrast, P-tyrosine and P-serine IRS1 show pretty small but consistent differences at all age brackets. Therefore, the previous evidence suggests that individual differences may exist in patients that have the same disease depending on their age. This is a way of stratifying individuals for when we want to enroll them in a clinical trial by age, and therefore we may hypothesize, have targeted hypotheses that those that are older will show changes in phospho-181 tau, and those that are younger will show changes in phospho-231 tau. So age stratification is something important. How about genes? We have also evidence that individualized responses to treatments may be based on genotype, and that this may be revealed by neuronal EV biomarkers. We have conducted a study of neuronal EV biomarkers using samples from a clinical trial of intranasal insulin in Alzheimer's, which was conducted in collaboration with Suzanne Kraft, a pioneer in the intranasal insulin field. In this study, it was shown that 20 international units of intranasal insulin had a positive effect on cognitive performance in AD patients, in early AD patients. This change in cognition was quantified as change in ADAS-Cog. And we saw that change in ADAS-Cog, which is a scale of cognition in Alzheimer's, change in ADAS-Cog is tightly correlated with both P-serine and P-tyrosine IRS1 in E4, ApoE4 negative individuals, whereas there is no relationship in E4 positive individuals. This suggests, in other words, that ApoE status modifies the association between change in cognition and change in EV biomarkers in response to intranasal insulin, showing target engagement in one genetic subgroup and not in the other. Our efforts in biomarker discovery and validation have brought us to a point where we now have actually a whole panel of biomarkers that assess amyloidosis, tauopathy, neurodegeneration, white matter hyperintensities, synaptic abnormalities, neuroinflammation, metabolic abnormalities, mitochondrial abnormalities, and neurotoxicity. And we hope in the future to analyze large cohorts measuring all these mediators and try to identify individuals that show preferential abnormalities in each one of these pathologic aspects of the disease. I will skip this slide and I will jump to conclusions to leave time for more discussion. Neuronal EVs contain biomarkers that reflect different aspects of disease pathogenesis, potentially resulting in multidimensional patient characterization. In relation to insulin resistance, which I specifically selected as a focus for this talk because it's not as widely appreciated and as amyloidosis or tauopathy, EV biomarkers suggest that insulin resistance underlies the pathogenesis of Alzheimer's, Parkinson's disease, other neurodegenerative disease, and multiple psychiatric diseases. And since it underlies so many diseases, it may not be causal to any particular of them, but it could be a non-specific marker of brain aging or vulnerability to stress from neurodegenerative cascades. Future directions are manifold, and we're already pursuing many of them. First of all, there is continuous need for methodological innovation. I am a firm believer that jumps and advances in science follow advances in technology and availability of better methods. We're already pursuing FACS studies, FACS sorting studies, cryo-EM applications to demonstrate EVs with accuracy, and improvement in the EV isolation methodologies to get an even cleaner and even more representative isolate of extracellular vesicles of brain cell origin. We have expanded our efforts in biomarker discovery, and we soon hope to have lipid biomarkers, RNA biomarkers from EVs, as well as new functional assays that will show how these EVs behave when are uptaken by cultured cells. The neuronal EV biomarkers can be used to select subgroups for clinical trials to demonstrate target engagement and predict treatment responses. So far, I have shown you how we can do that by leveraging already conducted trials and leveraging their samples. We are now in the process of conducting trials and collaborating with individuals where they have explicitly designated exosome biomarker as primary biomarker outcomes in their clinical trials, and we're eagerly anticipating these trials to be complete. Precision medicine clinical trials is the holy grail for us, where the right intervention will be applied to the right patients at the right time of their disease, and responses will be evaluated using the right outcome, which in our case will be exosome biomarkers. And I would like to acknowledge the hard work, imagination, and competence of my lab staff, postdocs, brilliant postdocs, postbacks, and regular staff members. I have multiple intramural, extramural collaborators in the US and various countries. It's an omission, actually, that I have omitted my Greek collaborators from this slide. I apologize for that. And special thanks to my mentor in the EV field, Ed Getzel, who pioneered these studies of neuronal EVs. And with that, I will stop sharing, and hopefully we'll have a discussion.
Yeah, Dimitris, thank you so much for the excellent presentation. It was a nice overview of the potential actually of EVs, not only in Alzheimer's disease, but also in other neurodegenerative diseases. So I will open the floor for discussions. As I said in the chat, whoever wants to make a question, please write your name in the chat or the question. So I have a first question from Fedon Brotsakis. So let me see. So Dr. Brotsakis asks, does the presenter think phosphorylation of residues mentioned in the case of tauopathies promote the formation of tau fibrils? If so, does he have any insight on which stage of tau aggregation or tau function this occurs? And I want to mention also that Dr. Brotsakis, if you want also to add any additional comments, please free to do so.
This is a fantastic question, and jumps right in something we're very interested in pursuing now. My thinking that is mostly reflects the publications by Mark Diamond and his group. I believe that what promotes aggregation of tau into fibrils is actually the 3D tertiary structure of tau, which can be facilitated by having some phosphorylations, but bad forms of tau can be facilitated by having phosphorylations in some areas. It is regarding exosomes, it is actually a question whether inside exosomes there are tau fragments or whole length tau. In collaboration with Dominic Walsh and some others at Harvard, we have shown that actually exosomes can contain whole length tau. And the recent paper in Brain by Tsuneya Ikezu and others showed that exosomes are very effective in propagating tau pathology to recipient neurons. But this is really points to the fact that exosomes are not just carrying these molecules, but they may be a mechanism for spreading the disease. I believe that exosomes can have whole length tau with bad phosphorylations that when uptaken by recipient neurons can act as a seed to promote the formation of misfolded tau in recipient cells.
And also, if I can add one small point about the precision medicine sort of perspective. So usually, so when there is a huge problem when we want to trace basically, the type of stage at which one is in the disease. So for instance, depending on the age and so on. So I was wondering, I mean, you put forward the approach of the biomarkers, but there is also another approach where you can, for example, design tracers, the so-called PET tracers. There are some for some type of fibrils, but there are none for other fibrils. So I was wondering if in this precision medicine approach, you're thinking along the lines of designing tracers where they can basically tracked at what stage or fibril formation stage you are, or oligomer formation stage you are, and so on.
This is funny because this is the one slide that in the last minute I decided to omit, but actually it's exactly the question. So I will share again and show you what we plan to do. Sorry for that. So decades of research have been using CSF, but mostly PET biomarkers, as you mentioned, and have established a timeline where progressive A-beta accumulation starts 15 to 20 years before cognitive symptom onset, followed by progressive tau accumulation, followed by abnormalities in FDG PET, followed by MRI atrophy, and finally cognitive impairment. And these associations are tighter with cognitive impairment the closer we get. So what we plan to do in the near future is we want to demonstrate the association between our biomarkers with amyloid PET, tau PET, FDG PET, and MRI in progressive longitudinal cohorts. And this will enable us to ask the more interesting questions and questions that relate to the pathogenesis. So for instance, since EVs spread through, since tau spreads partly through EVs, we can ask the question, does tau in EVs show abnormalities before tau elevation in PET? When we can do that by getting a group of individuals that are amyloid PET positive, tau PET negative, and examine their longitudinal samples and see whether abnormalities in tau in EVs precede or coincide with those in tau PET. Or we could test the hypothesis that neuroinflammatory markers in EVs become abnormal before neurodegeneration, because there is a theory that neuroinflammation may be crucial in development of atrophy and is close to the final pathway. Or that EV synaptic markers is a very late synaptic degeneration is known to be closely correlated with cognitive changes. So by examining samples of individuals that have been characterized multidimensionally with various PETs and with various CSF and other blood biomarkers, we would be able to deconvolve all these curves and show what is the exact role of EV biomarkers. So we don't do PET studies, but we collaborate with the groups that conduct PET studies in large cohorts. And we will try to do that. And these are going to be pretty large studies too.
Thank you.
Thank you, Dimitri. Next question is from Yannis Sotiropoulos. Yannis, do you want to make your question, or I can read it by myself?
Just go on. It's the same.
It's just no. You go ahead.
Okay, Dimitris, great talk. Really great to see the progress and all the development of these longitudinal studies you're conducting. Just fast, how you see, because I see more and more from your work and others that insulin signaling is more involved. What is your view from the clinical aspect? Because you can combine both molecular and clinical aspects in view on that, seeing patients yourself. How you see the connection between insulin signaling, the obesity, and depression with Alzheimer's disease? Because this is another triangle that not a lot is characterized, or even very poorly characterized. I mean, we are checking the stress, but however, stress and insulin signaling, we have discussed this in the past, is connected. But how about obesity? And so what is your view on that?
I think that actually there is peripheral insulin resistance, which is insulin resistance in tissues throughout the body. There's brain insulin resistance. These two correlate, but they are not identical. So you can have brain insulin resistance even in the absence of peripheral insulin resistance. And then there is neuroinflammation and peripheral inflammation, which are again correlated. I think that the emergence of insulin resistance in the brain is closely related to neuroinflammation. as well as the peripheral insulin resistance is associated with peripheral inflammation. I think there is definitely, there are epidemiologic studies that implicate peripheral inflammatory states and obesity and the emergence of mood disorders in particular. And so I think there are connections all along. I think that also sex is an important factor. And it's probably a subset of individuals that show develop depression as a result of inflammation and insulin resistance. It may not be everybody, but it is a good subset, as well as bipolar depression. And with biomarker studies, but you have the problem is that you have to look for something in order to find it. So in the past, perhaps psychiatrists were not looking so much for metabolic abnormalities in their depressed patients. But now they do. Now an increasing number of psychiatric research is focused on metabolism and looking for metabolic factors, obesity, neuroinflammation. And I think we will learn more and more about it.
Thank you, Dimitris. We have another question from Dr. Skarmeas. Nico, do you want to go ahead?
Okay. So great talk, Dimitri. I don't know, maybe you already touched on it because they were calling me. I'm still in the hospital. I was in and out. And do you have any insight on the relation with the new, quite promising plasma phospho-tau 217 or not? Because I saw slides, there was something about, yeah.
Fantastic question. This is kind of an elephant in the room whenever I give a talk now these days. So, well, it's not only 217. In this last summer, there have been phospho-181 tau assays, both a CIMOA and a mesoscale platform that showed amazing performance in distinguishing AD patients versus controls, and both prospectively, as well as 217 tau. These are very promising, and they have the potential of being very simple. So I'm open to the possibility that it could be, they could do the trick just as well or perhaps better than exosome biomarkers could do. Exosome biomarkers are laborious. And it could be that if a simple plasma assay does the trick just as good, what we plan to do is that we conduct large studies where we're going to actually implement one of these new promising assays in plasma, as well as exosomes. Only head-to-head comparison will be fair in this thing, in answering the question, what do we need to do? But I think that exosome biomarkers will go beyond the simple AD, non-AD. I think that their future is in precision medicine and in clinical trial domain, because one assay will tell us, okay, it may be good for clinical practice to distinguish in future AD patients from controls, but it's not gonna answer questions about what particular pathology may be more prominent in this person, or which pathology should we target in this particular person. So,
yeah, great.
That is my view.
Yeah, thanks. So we have.
But it may very well be the case, though, that we've been doing all this work all this time, which is pretty laborious, difficult to do all the exosome isolations, and the answer would be much simpler with the new technology in plasma.
Yeah,
it sucks, but we'll have to live with it and make the best out of it.
But there are all these mechanisms you described, right? So
exactly
that may define different subgroups. So I mean, you will have to say that it will be able to find very simple tests in serum or plasma for all these underlying mechanisms. So I don't think that's quite likely. Maybe we succeeded with tau, but who knows whether we succeed with the other ones.
I have to say, I have been surprised that a single marker has been showing so good performance in these studies. I was surprised to see, but these are legitimate studies, large groups. We have to accept what is published.
Great. Thanks.
Thank you. Dimitri, we have another question from Dr. Zaganas. Yanni?
Hello.
Hi, Yanni.
Thank you. Hello, everybody. Thank you for the great talk. So, based on the data you presented, do you think there's a promise in drugs for use for diabetes currently that they could be used also for AD?
Yes, and this is something that in part has been tried and in part will be tried also in future ongoing in future clinical trials. So one simple way is to give insulin. And this is what the studies of intranasal insulin have been trying to do. If you give insulin through an IV injection, you will cause hypoglycemia to a person who doesn't have diabetes. The person will fall down on you, so that you cannot do. But you can give intranasal insulin with the premise that this goes directly into the brain, and that's what Suzanne Kraft and others have been doing. Unfortunately, the trial results from the larger trials so far have not been positive. Another one that has been tried is pioglitazone. It's another type of diabetes medication, tried and failed. And then there are the class of GLP-1 agonists. Of those, liraglutide was recently tried in a big trial in England and had the primary outcome didn't show any change in cognition, but then there were some beneficial changes in tau PET. So there is some promise there. And I know that there are more and more trials that are going to be done with GLP-1 agonists. So there are numerous diabetes drugs. Maybe some of them will do the trick. Maybe some others do not. But I think that really touches upon the precision medicine. When we do trials and we enrolling people based on whether they have Alzheimer's disease or not, well, a subset of them may have very real insulin resistance, but some of them do not have insulin resistance in their brain. And now we're bringing them all to the anti-diabetes drug, and then it doesn't work in the group, and we say it doesn't work for Alzheimer's disease. Well, it may very well work in those that they have insulin resistance in the first place. So you try to correct something that it's not there. That's what we do these days.
Thank you, Dimitris. I think we have one more question. Maybe it's the last one that I can have because it's already 10:02. So this is from, we have a different group joining from YouTube. So this is from Dr. Sofia Tsiara. Thank you for this excellent presentation. I would really be interested to gain some insight into the process of selection of these specific molecules for immunoprecipitation of peripheral neuronal leaves. So I think it's a question.
Currently we're using L1CAM and NCAM. These are two cell adhesion markers that are known to be expressed in neurons, although they are partly expressed in some other tissues. And we hope in the future to develop additional surface markers that will allow us an even tighter immunoselection, an even more enriched population. Part of it can be informed by proteomic studies. If you see that other proteins and the surface of exosomes may be they would need to be widely expressed on neurons. They would in neuronal EVs. So there can be a screening phase where you have EVs from different neurons and see what molecules they express on the surface of the EVs they release. But then you need also good monoclonal antibody, and the whole thing needs to work and to show that it performs better than L1CAM and NCAM. So I don't think that L1 and NCAM are perfect, but they are the best that we have identified today. We and others are trying to find additional or alternative molecules to target. The field is wide open for anyone who wants to try.
Thank you, Dimitris. Thank you. I would like, first of all, to thank you for the excellent presentation, but also thank all the participants that joined us in this first seminar, open seminar series. I would say that if anyone has any question, they can contact you or me for more information. And soon we will send more information about the second seminar series next month. Thank you all for the participation.
Thank you. Thank you, Vaso.
Thanks.
Thank you.
Thank you.
Thank you.